I know it looks like there's a ton on this slide, but trust me, after we get through it, you'll see that this is an organizational scheme that helps you to understand sort of how we think about transporting different substances based on the direction that we're moving them in, that ties in with the thermodynamics of transport, as well as the kind of substance that it is. And on this, we're also going to sort of highlight and draw attention to the different substances that we've actually already talked about moving, or substances that we are going to study in this learning unit. Okay, so just big picture, the first thing that we need to think about is the nature of our metabolite, okay? And then we're going to examine the direction that it's going, and if it's a process that requires energy, where do we get that energy from? So those are sort of the main three questions that we're going to address. And then the four main types of transport that we need to think about are passive mediated or facilitated, and then active transport, in which there's two flavors: primary active transport and secondary active transport. So truly, there's probably three transport mechanisms we need to think about: passive mediated, primary active, and secondary active. But this flowchart here is going to help us understand how we determine which sort of mechanism is in play here. And then also allow us to sort of think about some of the examples that we'll see in some future sort of videos from this learning unit. Okay, so I'm going to start up here and highlight the first thing that we need to do is think about the nature of our metabolite. Remember, what we're talking about here is transport across a lipid bilayer, generated again from phospholipids, and why we need to consider whether a substance is hydrophobic or hydrophilic is things that are hydrophobic or nonpolar are really not going to need any assistance. So things that are nonpolar or hydrophobic are going to be able to pass through that membrane by what we call non-mediated transport. Remember, a mediator is something that helps something, so non-mediated transport means we don't need any help from anything. They're going to freely diffuse through the lipid bilayer, which is why hydrophobic or nonpolar substances are going to pass through in this way. And for this reason, the only direction that we can sort of have these substances go is from high concentration to low. There's not going to be any way to really build these up because they freely flow through our lipid bilayer. So here's some things that we've talked about already. We've talked about oxygen. Now remember, we talked about the 1 millimeter rule with oxygen, right? We talked about it in the context of oxygen sort of delivery and ferrying between bringing it in when you take in a breath in your lungs, traveling across your alveoli and sort of getting into the hemoglobin that's bound in your red blood cells, transiting through your circulatory system, and then being offloaded ultimately to myoglobin that's embedded within your tissues. But the importance of not having to diffuse more than 1 millimeter is also related to the fact that when we think about this sort of going through and lipid bilayers, oxygen being relatively, again, small, nonpolar, we don't have to worry about any sort of transporters to facilitate that. Just a little side story here. Water used to be the same sort of thought process. It used to be thought that, well, water, the concentration is so high, it must freely flow. But we now know that there are transporters called aquaporins. We're not going to study those, but aquaporins that facilitate sort of the transport of water across lipid bilayers. Some other substances that we've talked about already: cholesterol. We do not have cholesterol transporters in the sense that it is a freely flowing hydrophobic molecule that can go through lipid bilayers. Importantly, we see that cholesterol is often embedded within those lipid bilayers. Okay. And then lastly, we're going to talk about a substance. We've talked about this already, but 2,4-dinitrophenol. Okay, we'll talk about how and where it's used. Just a reminder, we've talked about this molecule before. It was a diet pill in the 1940s, but we'll talk about why it's no longer a diet pill that's readily prescribed because of the downstream sort of consequences of it. So lots of things probably fall into this category, but they're not going to be the focus of our study here because we're interested in moving things often that are hydrophilic and that are going to need assistance then getting across lipid bilayers. So anything that falls into this bucket, we define as mediated transport. We need assistance. It's going to be facilitated by proteins. Remember when we learned and talked about lipid bilayers, we talked about sort of that fluid mosaic model. The idea that we've got this lipid sea where we have icebergs embedded in it. And those icebergs were really proteins. Those are the gatekeepers. Those are the transporters that are going to be facilitating the movement of things across the lipid bilayer. So mediated transport represents the class of everything that we sort of talked about, from passive mediated to primary and secondary active transport. So anytime we need some help moving our metabolite because it is something that is not nonpolar, it's hydrophilic, we need to have a protein mediator for that, okay? The next sort of decision point that we need to think about in terms of what type of transporter are we going to use is we need to think about the direction that the substance is going. And ultimately, it's going to be going with or against the concentration gradient. So that's sort of the defining feature. Are you going with or against the concentration gradient? If you're going with your concentration gradient, we're going to define that as downhill. So something that's going downhill is going to have a negative delta G. So again, downhill processes are going to have a negative delta G. We would define those as being spontaneous. Something that's going against the concentration gradient, we would define that as an uphill process. From a delta G standpoint, that's going to be a positive delta G. That's going to be non-spontaneous, right? So a non-spontaneous process, because of that positive delta G, is going to require energy. Okay, and we're going to get to what those pieces are, but coming back here, once we understand the direction that we want our substance to go, are we dissipating a proton gradient or building a proton gradient? That's probably a good word to sort of put in here. This is going to be dissipating. This is going to be generating. Those are words that we'll use. So whether we're generating a gradient or dissipating a gradient, we'll bring in these terms. So facilitated diffusion, or sometimes called passive mediated diffusion, we still need a transporter assistant. We need that protein to be able to help us. But since we're moving things in a downhill direction, we don't need to have any additional energy put in. We call this spontaneous process passive mediated or facilitated diffusion. So lots of examples of things that we'll talk about with this, just giving you a little bit of a laundry list of ones we'll talk about. We're going to be talking about GLUT2 and GLUT4 glucose transporters. We are going to be talking about porins, which are going to be used to transport carbohydrates in bacteria. Okay, we're going to be talking about an antibiotic called valinomycin. Okay, and we're also going to be talking about, actually, that's the only three we're going to be talking about that are going to be passive mediated in this way. So valinomycin, GLUT2 and GLUT4 receptors, and then porins, which are responsible for glucose transport in bacteria. I want to highlight that really valinomycin is all about transporting potassium. All right, let's go over on the other side here and highlight. I'm not going to give any examples yet because it is really not fully correct to say that the transport mechanism is active transport, right? Active transport requires energy. Where we have this final delineation is asking, well, what is the source of energy? So we know that it requires energy to go uphill, so where do you get that energy? And there's two places you can get that energy, and that's how we delineate primary versus secondary active transport. If you have ATP hydrolysis as the source of your energy, so ATP hydrolysis means that it's primary active transport. If it is another molecule's gradient, okay, another molecule's gradient is going to be secondary active transport. So again, all of these refer to active transport mechanisms, but again, primary active transport versus secondary active transport, the difference is what the source of the energy is. If you are burning your money directly, you're doing ATP hydrolysis, right? That is going to be primary active transport. And if you are using another molecule's gradient, that is secondary active transport. So examples of ones that we will see and talk about here, we are going to be talking about the sodium potassium ATPase. So sodium potassium ATPase is an example of primary active transport. We use ATP, so that name ATPase implies that we are burning up that primary currency in order to move both sodium and potassium cations against their concentration gradient. Secondary active transporter, the main one that we're going to be talking about is the SGLT, the sodium glucose transporter. Okay, that's responsible for transporting glucose, and it's going to use a sodium gradient in order to move glucose against its concentration gradient. Okay, so little summary here that gets us to think about the names of the different transporter systems that we need to think about. We need to think about facilitated diffusion, also called passive mediated. We need to think about primary active transport, and we need to think about secondary active transport. I don't have a specific slide on this, but I just want to put up here for us to think about within sort of these buckets of types of transport. We also have types of transporters in terms of the number of molecules that they're transporting and the direction if we are moving more than one. So I just want to write these down here so that we sort of have them down. We'll refer to them later. There can be a transporter that is a uniport. And like the name sort of means, we're only transporting one substance. Now you can have a primary active transport that is a uniport, okay? Or you can have a facilitated diffusion that is a uniport, okay? So the key here is that we're only moving one thing. If we are moving two things, we basically are going to have two possible options for ways that we can move two things. We can move them in the same direction. That is going to be a symport, or we can move them in the opposite direction. That is going to be an antiport. So uniport moving one thing, symport and antiports move two things. Symports move those two things in the same direction. Antiports move those two things in the opposite direction. So we'll make sure to clarify all of these transporters as we learn about them, whether they are facilitated diffusion, primary, or secondary active transport.